In mammals,
You and me, baby, ain't nothing but mammals.
sex determination is typically dependent on the inheritance of the sex chromosomes, X and Y. Individuals with two X chromosomes are genetically female, and individuals with one X chromosome and one Y chromosome are genetically male. It is actually only a single gene on the Y chromosome that determines whether an embryo develops as a male or female. This gene is known as the sex-determining region of the Y chromosome, or SRY for short. Or SRY for short.
Say you want to get rid of an invasive species, like get Asian carp out of the Great Lakes. All you have to do is release a gene drive that makes the fish produce only male offspring. In a few generations, there'll be no females left.
A research team from the Department of Animal Science and the School of Veterinary Medicine at the University of California, Davis, led by Dr. Alison Van Inenum and her graduate student Joey Owen, had an idea to create a bull with the SRY gene duplicated on the X chromosome. The researchers hypothesized that such a bull would only produce male offspring.
In a few generations, there'll be no females left.
As the SRY gene would be inherited on both the Y and the X chromosome. But how do you go about making such a bull?
Many of you have probably heard about CRISPR, so I'll just say briefly that CRISPR is a tool that allows researchers to edit genes very precisely and easily and quickly. It does this by harnessing a mechanism that already existed in bacteria. Basically, there's a protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. The result is basically a word processor for genes. You can take an entire gene out, put one in, or even edit just a single letter within a gene. And you can do it in nearly any species,
including our own species.
How do you get your trait to spread? This is where it gets clever.
The H11 locus on chromosome 17. This locus is a region between two genes and has been shown to be a safe insertion site for new genes.
I'm 17.
On chromosome 17.
The team therefore decided to include a reporter transgene in their donor template to confer an easily identifiable characteristic to allow for fluorescent screening of embryos to identify those carrying the insertion.
The newest undercover reporter.
A reporter transgene in their donor template.
They therefore included the green fluorescent protein, or GFP, alongside the SRY gene. This meant that any blastocyst with an SRY insert would also glow green when exposed to light of a certain wavelength.
Hey, that girl's great.
The technology to do this, to genetically engineer an organism and include a gene drive, is something that basically any lab in the world can do.